Welcome to today's deep dive. We are constantly told that, water's the absolute source of life. Drink your 8 glasses a day, stay hydrated, water is healing, all of that.
Right, it's the golden rule of health.
Exactly. But if we zoom in to a microscopic level, into the actual fluid bathing your cells, that same life-giving water is actually accelerating a relentless, invisible war against your DNA.
It is, it's one of the great biological ironies, honestly. The very fluid that keeps our tissues functioning also happens to be the perfect chemical conduit for one of the most destructive molecules in the human body.
Which is exactly why we're taking you, the listener, on a deep dive into this microscopic war zone today. I've got a massive stack of sources in front of me right now. We've got density functional theory studies, the Milligan and Ward research on cellular scavenging, plus a few clinical preprints.
Oh yeah, lots of heavy reading for this one.
For sure. And our mission is to distill all of this for you. We're going to explore the exact mechanisms of how a specific treatment, called polynucleotides, act as this ultimate microscopic bomb squad. We're looking at how they scavenge highly destructive free radicals to protect your most vulnerable fluid-filled tissues.
Tissues like your gut, your eyes, your respiratory tract, yeah.
Right. And we are going to look at the how and the why, because understanding how a microscopic bomb squad works well, it requires us to fundamentally understand the bomb they're actually trying to defuse.
Exactly. And looking at the research, the bomb in this scenario is something called the hydroxyl radical, which is a very specific type of reactive oxygen species, or ROS.
Now, to be fair to our bodies, ROS are actually natural byproducts of how we metabolize oxygen. We need oxygen to breathe, and the sort of metabolic exhaust fumes of that process are these reactive molecules.
Right. They aren't inherently evil, but when those exhaust fumes build up too fast, we enter a state called oxidative stress. And let's clarify what a free radical actually is, chemically speaking, because it sounds like a buzzword.
Yeah, let's unpack that.
Fundamentally, it's an unstable molecule that's missing an electron. And because it's incomplete, it is incredibly aggressive. It will violently tear through your cellular machinery, ripping electrons off of lipids just to destroy your cell membranes or, modifying proteins so they just collapse.
But the sources say the hydroxyl radical is unique, right? Because it bypasses all of the surface level stuff and goes straight for the instruction manual. It specifically targets your DNA.
Yes, and the density functional theory studies, or DFT studies that we have in our stack, map this out perfectly. They let us see the precise quantum mechanics of this attack.
Yeah, I was looking at those. They show that this hydroxyl radical loves to target a very specific part of the DNA structure. It goes after the amine group, the NH2 of 2 specific DNA bases, guanine and adenine.
I'm really glad you brought up the DFT studies because guanine and adenine, they have an electron distribution that effectively paints a giant target on their backs. The hydroxyl radical comes in and physically abstracts, meaning it violently rips away a hydrogen atom from that DNA base.
Just to satisfy its own missing electron.
Exactly. And once your DNA loses that hydrogen, the structural integrity is compromised, which leads to dangerous mutations.
And here is where we get back to that biological irony about water I mentioned earlier, this violent hydrogen theft. It isn't happening in a dry vacuum. The DFT studies actually compared this reaction in a vacuum versus in water.
Right, a process called solvation.
Yeah, solvation. Can you explain how that actually works?
Sure. So water molecules essentially surround the target. And because of water's unique hydrogen bonding network, it actually lowers the energy barrier for the chemical reaction.
So it basically greases the wheels.
It absolutely does. Water acts as a chemical catalyst that dramatically accelerates the hydroxyl radical's ability to abstract that hydrogen atom.
Okay, wait. Let's unpack this for a second, because this seems like a massive biological design flaw. Aren't our mucosal cells, like the linings of our eyes, our lungs, our gut, aren't they basically swimming in fluids?
Oh yeah, completely bathed in them.
Are you saying they are at a biological disadvantage simply because they're wet?
It is a massive vulnerability, yes. Mucosal tissues are prime targets for oxidative stress precisely because they are highly hydrated. And if we look at how this plays out in real life, you can see this exact vulnerability driving incredibly common diseases.
Right. Like if you've ever dealt with severe dry eye, you've experienced this firsthand.
Oh, absolutely.
The sources point out that when tears evaporate too quickly and become too salty, which is clinically called hyperosmolarity, It triggers a massive production of these reactive oxygen species in the eye.
And because the eye is a fluid environment, solvation accelerates those radicals, allowing them to rapidly mutate and destroy the corneal cells. We actually see the exact same mechanism in the lungs, too, particularly with asthma or severe respiratory infections.
Because the airway is lined with fluid and mucus, right?
Exactly. When oxidative stress hits the lungs, it causes the airway mucins to chemically cross-link. It turns a healthy, flowing fluid into a rigid, sticky trap. And as the cells lining your lungs die from these radical attacks, they burst open. Yeah, they burst and release their own DNA right into that trap. This is what forms those incredibly dense, dangerous mucus plugs that physically block airflow in asthma patients.
That's wild. And let's ground this in something almost everyone listening has probably done, which is taking a painkiller on an empty stomach.
Oh, yeah. Classic mistake.
Common NSAIDs like indomethacin or even just regular ibuprofen are notorious for causing gastric ulcers. And reading these sources, it makes total sense now.
It really does. Those drugs spark a huge surge of free radicals in the highly fluid, highly acidic environment of your stomach. They literally ignite a localized war right into the tissue.
So your mucosal cells are sitting ducks, bathed in the exact fluid that speeds up the bombs trying to destroy them.
Yeah.
Which begs the question, how do we not just dissolve? Like, how do these tissues normally survive this constant onslaught?
Well, they rely on a highly coordinated natural defense system. It starts with physical barriers. In the gut, for example, your body expresses something called the MUC2 gene.
MUC2, right?
Yeah, and this gene manufactures a massive gel-forming mucin. By weight, this mucin is actually 80% complex sugars or oligosaccharides. It forms an incredibly thick, insoluble physical shield over the vulnerable gut lining.
So it's essentially the castle wall. And behind that wall, we have chemical defenses, right?
Yeah.
Things like prostaglandins and nitric oxide that maintain blood flow to the tissue and keep bicarbonate levels high enough to neutralize stomach acid.
Exactly. And we also have our own endogenous antioxidants. These are internal enzymes with names like glutathione, superoxide dismutase, and catalase.
And what's their job in all this?
They constantly circulate through our fluids, willingly handing over their own electrons to the free radicals to pacify them before they can reach the DNA. But here's the core problem. Under severe oxidative stress like from that NSAID or a severe infection, these natural antioxidants get completely depleted.
The defense budget basically runs out.
And defense budget completely runs out.
And when it runs out, we hit something mentioned in the Milligan and Ward study called the clustering paradox. This was a huge aha moment for me reading the sources.
It's a really fascinating concept, isn't it?
It is, because you would logically think that even if your body can only scavenge, like some of the free radicals, that's still better than nothing. But the research shows that if your scavenging capacity isn't perfectly complete, it can actually backfire on you.
Yeah, the clustering paradox is a terrifying quirk of cellular biology. If your biological environment can only scavenge, say, 50% of the radicals, the remaining 50% don't just spread out evenly.
They cluster up, right?
Exactly. Due to the complex physics of how they diffuse through the tissue, they tend to cluster. They group together and strike the cellular DNA in a very tight, localized area.
So it's like getting a single scratch on your car versus taking a dozen deep dents to the exact same spot on the door. A single hit is an easy fix. The body's repair shop handles it. But a cluster just completely overwhelms the system.
I would actually take that car analogy a step further. It's not just that the dents overwhelm the mechanic, it's that the tools the mechanic uses physically cannot fit into a clustered dent.
Wait, really? The repair enzymes literally can't fit?
Yes, physically.
Yeah.
In a cell, when you have multiple radical hits in a localized cluster on the DNA strand, the cellular enzymes responsible for repairing it get in each other's way. The repair machinery physically stalls out. A clustered lesion is repaired exponentially more slowly than a single, isolated hit,
Leaving the cell totally vulnerable to permanent mutation, or just cell death. So the castle wall of the MUC2 gel is breached, the antioxidant defenders are dead, and clustered bombs are destroying the DNA instruction manual. The cell is losing the war.
And that is exactly when it needs outside reinforcements. It needs a specialized intervention that can bypass those depleted natural defenses. And this is where we introduce polynucleotides, or PNs.
Okay, let's unpack exactly what these are, because the sourcing here is kind of wild. Polynucleotides are biopolymers. They're literally chains of nucleotides, which are the fundamental building blocks of DNA.
Right.
And to get these highly purified fragments, scientists use a process called PNHPT, or high purification technology. And they extract this DNA from the gonads of salmon trout.
I know, I know. It sounds like science fiction or perhaps just bizarre, but there is a profound biological logic to it.
Please explain the logic, because I was definitely scratching my head.
Well, reproductive tissue, like gonads, is incredibly dense with DNA. It's essentially nature's concentrated storage facility for genetic material. And salmon trout DNA has a surprisingly high degree of biocompatibility with human cells.
Because the HPT purification process strips away all the fish proteins and fats, right? So your body doesn't reject it.
Exactly. The process removes anything that would trigger your immune system to attack it. What you're left with are pure, naked chains of DNA. They're highly stable, they resist extreme temperatures, and they have a very specific ability to absorb UV light at 260 nanometers.
But their most important feature is how they act as direct chemical scavengers, right? Let's talk about the mechanics there. How does injecting highly purified fish DNA stop a hydroxyl radical from destroying my gut lining?
Remember earlier when we defined a free radical? We said it's desperately seeking an electron, and it usually violently steals it from your cellular DNA.
Right, by abstracting that hydrogen atom.
Exactly. Well, polynucleotides are packed with nitrogenous bases, which means they are absolutely loaded with easily accessible electrons.
Oh, I see where this is going.
Hmm.
So when the hydroxyl radical comes tearing through the fluid, ready to attack your DNA, it bumps into this massive cloud of polynucleotides first.
Precisely. The PNs act as sacrificial electron donors. They readily hand over their electrons to the hydroxyl radicals, pacifying them chemically.
So the radical is just neutralized instantly.
Yes, it halts the chain reaction of destruction before it ever reaches the host cell's actual DNA.
They're essentially sacrificial decoys. That makes so much sense. We're flooding the war zone with highly purified fish DNA, so the free radicals shoot at the decoys instead of our vulnerable cells.
Exactly.
And that completely bypasses the clustering paradox, because the bombs are being diffused out in the fluid rather than on the cell surface where the repair tools can't fit.
That decoy mechanism is the primary physical defense, yes. But what elevates polynucleotides from a simple chemical sponge to a highly sophisticated biological therapy is what happens next, because they don't just take the hit. They actively trigger a massive cellular counterattack.
Yeah, this is where the sources transition from chemistry to pharmacology. Once the immediate threat is neutralized by the decoy, a specific type of polynucleotide formulation called PDRN interacts directly with the surface of your cells.
Right, it acts as an agonist.
Specifically for something called the adenosine A2A receptor. It binds to this receptor and turns it on like a biological key in a lock.
And turning that key initiates a really profound cascade of healing inside the cell. When the A2A receptor is activated, it signals the cell to produce more of a messenger molecule called CAN-MP.
Let's translate some of this cellular alphabet soup for the listener, because the research papers are incredibly dense with acronyms here. What does increasing this CAN-MP messenger actually achieve?
Think of CAN-MP as the cell's internal emergency broadcast system. When its levels rise, it sends a signal to suppress the cell's master switches for inflammation, specifically pathways known as NF-κB and MAPK.
So by shutting down those pathways, the cell instantly stops pumping out the chemical alarms that cause swelling and redness.
Exactly, the pro-inflammatory cytokines drop.
Yeah, the sources note that destructive signals like TNF-alpha, IL-1 beta, and IL-6 all plummet. And at the exact same time, the A2A receptor tells the cell to release IL-10, which is an anti-inflammatory signal.
It's flipping the switch from destroy to rebuild. It is a total paradigm shift for the cell.
And the rebuilding phase is incredibly robust. Activating the A2A receptor stimulates another crucial biological circuit called the Wnt beta-catenin pathway.
Yes, the Wnt pathway.
I was reading about this pathway, and it's like the cellular foreman. When it gets activated, it starts reading the architectural blueprints of the cell to drive active tissue repair. It tells the cell to stay alive by suppressing death signals, and it starts the process of neurogenesis.
Literally growing new nerve endings in severely damaged tissue, yes. The foreman analogy is highly accurate, and, every good foreman needs building supplies.
Right.
This is perhaps the most elegant part of the entire polynucleotide mechanism. We talked about how the PN decoys get chemically shredded by the free radicals taking the bullets for us, right?
Yes, they get destroyed in the crossfire.
Well, those shattered pieces of fish DNA don't just float away as waste. Polynucleotides prime what is known as the salvage pathway. The host cell literally sweeps up the broken free-floating nucleotides from the destroyed decoys and internalizes them. It's true. It uses them as free, raw biological bricks to synthesize its own new human DNA.
That is the ultimate microscopic recycling program.
It truly is. The cell takes the debris from the battle and uses it to rebuild. The research shows this process increases the expression of the p53 protein, which coordinates DNA damage repair, and it massively boosts mitochondrial DNA replication.
Which gives the cell the sheer energetic horsepower it needs to rebuild its extracellular matrix, the ECM scaffolding that holds the tissue together. And at the same time, it suppresses the degrading enzymes, like MMPs, that would otherwise chew that scaffolding up.
Exactly.
So to recap the bomb squad so far, we've got the sacrificial decoys taking the hits in the fluid. We've got the A2A receptor acting as the medical team turning off the inflammatory fire alarms. And we've got the salvage pathway recycling the shattered decoys into fresh building supplies.
It is an extraordinarily comprehensive system on its own, but the clinical preprint studies in our stack reveal that polynucleotides actually have a partner.
A party.
Yes, and when you combine them, you don't just get an additive effect, you get the ultimate synergy. That partner is hyaluronic acid, or HA.
Okay, now, I really have to push back here for a second. When I hear hyaluronic acid, I think of the little serum bottle sitting on my bathroom counter. It's a standard moisturizer for skincare.
That's what most people think, yes.
Are you telling me that a basic hydrating serum is actually being used as a tactical defense mechanism inside the body?
It is vastly more than a simple moisturizer, especially when we look at the physics of the microscopic war zone. Hyaluronic acid is a massive, heavily charged molecule. While it does possess a very minor ability to scavenge free radicals on its own, its true superpower is its exceptionally high water-binding capacity.
Right, it holds an enormous amount of water. But wait, earlier we established that water is the enemy. We talked about the salvation effect, that water actively accelerates the free radicals' attack on our mucosal cells.
Yeah.
How does adding a molecule that traps more water actually help?
That's the genius of it. HA doesn't just attract water, it fundamentally alters the physical viscosity of the fluid environment. It creates an incredibly dense, viscous, gel-like microenvironment.
Ah, okay. So instead of the free radicals tearing through a loose, watery fluid at lightning speed, they're suddenly trying to move through a thick gel.
Exactly. It's like trying to run through quicksand. The dense HA matrix physically dilutes the reactive oxygen species and drastically slows their diffusion rates across the tissue. They literally cannot travel as fast.
And because the radicals are bogged down in this molecular quicksand, the polynucleotide decoys have significantly more time to interact with them, donate their electrons, and neutralize the threat.
You've got it. It's altering the physical physics of the battlefield to give the bomb squad the upper hand.
That's brilliant. And the experimental data backing this up is pretty striking. The sources reference a study where human dermal fibroblasts, skin cells, were exposed to massive doses of UVA radiation.
Which generates huge amounts of oxidative stress.
Right. But when they treated the cells with a combination of HA and polynucleotides, they saw a synergistic effect, meaning 1 + 1 didn't just equal 2, it equaled 3.
Yes. The HA and PN combination didn't merely lower the free radical levels back to a normal baseline. It synergistically instructed the Wnt pathway, our cellulophorin, to massively upregulate the specific genes responsible for pouring fresh collagen and fibronectin.
The COL1A1 and FN1 genes, right?
Exactly. Essentially hypercharging the structural repair of the tissue.
And didn't it also upgrade the cell's own internal defenses? Because that part stood out to me.
It did, and clinically, this is perhaps the most impressive outcome of all. The HA and PN combination didn't just act as an external shield. It actually upregulated the cell's own endogenous antioxidant enzymes.
It forced the cell to produce more of its own internal bomb squad enzymes like GPX1 and SOD2 to prepare for the next attack.
Precisely. It neutralized the immediate threat, recycled the debris to repair the damage, and then trained the cell's internal army to be stronger for tomorrow.
That is just incredible. So, to bring this entire journey together for you listening, we started with a hydroxyl radical, this unstable little bomb missing an electron, ripping through the very fluids that give us life, using water as a catalyst to steal hydrogen atoms and mutate our DNA.
Right, and we saw how our natural mucus barriers like the MUC2 gel and our internal antioxidants can get completely overwhelmed by everyday things like painkillers or dry eyes.
Which leads to that terrifying clustering paradox where the DNA repair machinery physically stalls out because the damage is just too dense. And then we explored the microscopic reinforcements. Highly purified, biocompatible DNA fragments harvested from salmon trout.
Polynucleotides acting first as a sacrificial decoy, flooding the area with easily accessible electrons to pacify the free radicals before they ever reach our cells.
Followed by their role as a pharmacological key, activating the A2A receptor to shut down the swelling, start the neurogenesis, and initiate the salvage pathway to recycle the decoy DNA into human tissue.
And finally, pairing that fish DNA with the quicksand of hyaluronic acid to alter the physical viscosity of the battlefield, slowing the free radicals down and giving the decoys time to work.
It's just a perfect synergy. It's not slapping a Band-Aid on a symptom. It's treating the absolute root cause of oxidative stress to return the vulnerable cells of your gut, your eyes, and your lungs back to true homeostasis.
It is a remarkable, multi-layered approach to cellular defense that really bridges the gap between physics, chemistry, and pharmacology.
It really is. And it leaves me with one final kind of mind-bending concept for you to mull over as we wrap up today. We've been talking about using this technology to heal damage that has already occurred, right? To step in when the body is actively losing the war.
Yes, as a reactive treatment.
But if we can safely use exogenous purified DNA fragments as sacrificial shields and architectural blueprints, could the entire future of medicine become preemptive?
That is the logical and honestly incredibly exciting next frontier in this field.
Imagine a future where we don't wait for the ulcer to form from the NSA dudes, or we don't wait for the asthma plug to choke the airway, or the dry eye to mutate the cornea. Imagine a reality where we routinely preload our vulnerable mucosal barriers with this decoy DNA.
We could deploy the microscopic bomb squad before the bombs ever drop.
Exactly, giving our tissues a permanent molecular quicksand shield. We could theoretically make our bodies practically bulletproof to the oxidative stress of daily modern life before disease ever has the chance to take root. And that is definitely something to think about. Until next time, thanks for taking the deep dive with us.